Thanks to an acquisition that we completed just over a year ago of Jade Therapeutics, EyeGate now has two unique proprietary platforms in-house. One of them is based on, which we acquired from Jade, is based on cross-linking hyaluronic acid, and the other one is based on iontophoresis, which is our legacy technology. Both of these platforms have a product in the clinic, and both of them are late stage, and we expect to do our first filing with each of these platforms towards the end of this year, early next year. So the first one, hyaluronic acid. So hyaluronic acid, it's endogenous to the body. It does a lot of tasks in the body, but two of these we believe are ideal for the ocular surface. So we find it in the derm space, and we see that it promotes wound healing. We also see it in the synovial fluid, and obviously it's a natural lubricant. So these two properties we believe are ideal for the ocular surface. So what we've been able to do is we've been able to formulate with cross-linking a nice liquid gel or topical formulation, and that's what we have in the clinic. We see that hyaluronic acid's already been approved in the US. We see it in the derm space, and we see it in osteoarthritis. We see that it's also been approved as a topical formulation outside the US, in Japan, Europe, and other countries. When we talk about what we're able to do, though, just differentiating from straight HA, with this cross-linking, we're able to prevent degradation. So one of the biggest problems HA has is that it degrades rapidly. In fact, about a third of it degrades or turns over every 24 hours in our body. So being able to resist or prevent the degradation is really important. The way we formulate it with this cross-linking also allows it to stay on the ocular surface for long periods of time. So we say it's staying there for about an hour and a half to 2 hours, and it does this without causing any blurriness. So now we've got this nice eye drop that stays on the surface of the eye for about an hour and a half to 2 hours. It's a natural lubricant, so it feels good, and it's also promoting wound healing. So there's a lot of things we can do with that. So the first clinical study that we've gone into is large epithelial defects based on patients that have undergone PRK. So we did a very simple study. It was 3 arms. The first arm was just our eye drop, 4 times a day for 2 weeks. The other arm was our eye drop in combination with a bandage contact lens, and the third arm was just a bandage contact lens on its own. So we wanted to understand if we could actually accelerate the reepithelialization of the corneal wound. And we were able to demonstrate that. So it was a small study, as I mentioned, about 39 subjects, and we were able to demonstrate fairly powerfully with this small study that we can actually accelerate the closure or the wound that occurs from PRK. So what you see with the data here at the bottom of the chart is that we had 12 subjects in just the eye drop arm alone, and we had 13 subjects in the BCL or bandage contact lens arm alone. And you can see that we had about 30% more subjects or patients with a full closure at day 3 than we saw in the BCL arm. Okay? We also saw a much smaller wound size, 24 hours, and also at day 3 than you would see in the BCL arm. So we're pretty excited about this. We're actually just filed an IDE today so that we can continue on with our next study. It's going to be a similar design. We're going to be enrolling 45 subjects. There'll be 3 arms. But in that study there, we're going to have 2 arms which are just the eye drop alone. But instead of four times a day, we're going to have 1 arm where we do 8 times a day for 3 days and then drop down to 4 times a day. And then the third arm will be BCL. All right, so we're excited about that, but we're also excited about what this can be doing outside of large epithelial defects created by PRK. So there's a lot of other things that we think that this product could be used for. There's a lot of epitheliopathies or corneal defects that occur where it would be great to have a product like ours that could go on the eye and promote acceleration or reepithelialization. So we think there's a large market opportunity for this. This is an eye drop. This is very easy to use, and we're happy to show that we're going to be through the clinic, hopefully by the end of this year, and filing, if not by the end of this year, early next year. And it's going to be filed as a 510(k) de novo product. Okay, so we're looking forward to that. The other platform is the iontophoresis platform. So this is based on like charges repel. So we need to put a charge on the drug. We put a charge on the drug, we can deliver it. The first product that we're delivering is a corticosteroid. And we're actually developing this for 2 different indications, uveitis and cataract surgery. And I'm excited to say that we've been able to partner this out with Valeant, Bausch & Lomb, who've been amazing partners with us on both of these indications. And we're looking forward to filing we're getting ready to file right now to move forward with the protocol for the phase 2 or the second study for cataract surgery. Okay. So all I show here, I'll skip over to cataract surgery. We've done our preliminary study in cataract surgery. We did 80 subjects. What we were looking for is dose ranging. So we were looking for the ideal iontophoretic dose to use and the ideal regimen that we would want to use. The idea here, though, is they get a treatment with iontophoresis by the doctor in his office, and no drops are required. So we did 8 different cohorts with 10 subjects, and we found a couple different iontophoretic dosages that worked really well. We've chosen one of those to move forward, and we're looking to start that study in the next couple of months. Lots of catalysts, because we're looking to file with that product there. We're looking to file for anterior uveitis towards the end of this year, early next year, and do a supplemental NDA for the cataract surgery, 6 to 8 months later. All right, and that's the summary and the story. Thank you, everyone.